next what we are going to see is the other type of column chromatography, which is partition column chromatography. So the first one was adsorption column chromatography. So if adsorption has to taking place, the stationary phase has to be solid. Here we are going to see what is partition column chromatography. So here also we use a column of glass or steel, but the principle of separation is partition. So partition means there is no point for solid. So it could be either liquid gas or liquid chromatography. So here, for example, we will see is liquid partition chromatography. So that means the stationary phase and the mobile phase both are liquids in this example. Now, so the first question you may ask is how a liquid can be a stationary phase, because liquid is flowing. Liquid cannot stand by itself. So how a liquid can be made as a stationary phase. So what here we will do is we will take, for example, this green colored stuff, that is your column. So this is inner walls of your column. So either you can coat a liquid phase on the walls of this column. So that's what we call the wall coated open tubular column. So you are coating your liquid phase on the walls of this tube. So that is wall coated open tubular column. The other one is you will first coat a solid material on the walls of the tube, and you will impregnate that solid material with a liquid phase. That means you are actually coating a solid phase on the tube first, inside the walls of the tube first. So that is a support, and you will coat your liquid phase on that solid phase. So that is what we call the support coated open tubular column. So these are the two ways, how a liquid is made as a stationary phase. Okay. Now the mobile phase is a liquid in this case. So this is a liquid partition chromatography. So the partition is the principle of separation. We will see what is partition later. So in liquid partition chromatography, the stationary phase is a thin layer of a liquid held on the surface of a porous inert solid. Support, for example, you can use cellulose, silica gel, starch, calcite, kieselguhr. It's a kind of mineral. So these are the solid support which we will coat on the walls or inner walls of the column. Then we will coat our liquid phase on this solid support. So that will form the stationary phase. The mobile phase is a liquid, very important, which is immiscible with the stationary liquid phase, because in stationary phase we have a liquid phase. So your mobile phase should never miscible with the stationary phase. So these are two immiscible liquids with and that forms the mobile phase. And the principle is different components of the sample mixture have different partition coefficients for a given pair of immiscible liquids. So partition is, we will see what is partition in the next slide. So the partition coefficients are different for the different components in your mixture. So based on that difference, your mixture the components get separated when it passes through the stationary liquid phase. So what we will see what is partition coefficients. The partition coefficient K is given by K equal to Cs divided by Cm, where Cs is the concentration of a component in the stationary phase, and Cm is the concentration of the same component in the mobile phase. So it's a ratio between the concentrations. The concentration in the stationary phase divided by the concentration of the mobile phase. That ratio, what we call the partition coefficient, that is different for different components in your mixture. So that is the reason for the separation. So the component with the highest K partition coefficient is retarded most by the stationary phase and get eluted as the last component. So that is very clear, because the component with the highest K means what? Because K is Cs divided by Cm, where Cs is the concentration of your component in the stationary phase divided by Cm is the concentration of the component in the mobile phase. So if the K value is higher, that means the numerator is higher than your denominator. Okay. So what is numerator? Numerator is concentration of your component in the stationary phase. So that means a higher K value means your component prefer to stay in the stationary phase most of this time than in the mobile phase. So that means your component will retarded most. The component which has the highest K value will be retarded most, and that will eluted as the last component. So that will come at the last component as the last component at the end of the column. Okay, so this is how the separation of the components in your mixture happening in liquid column chromatography. So if you see what are the applications of liquid column chromatography, means it has a higher reproducibility than adsorption column chromatography and yields sharper and more symmetrically shaped bands. The next one is you can use this technique for the quantitative separation of components in a mixture of organic or inorganic. Even you can separate inorganic components by using liquid partition column chromatography. For example, if you take, you can separate cobalt and nickel ions by using this technique, and you take water supported on cellulose. So this is your stationary phase. You coat water on a cellulose, then acetone as cellulose, acetone containing some HCl. So this, if you can use water support on cellulose, and the mobile phase is acetone containing some HCl substances. So if you take this system, you can separate cobalt and nickel ions. So that means liquid partition chromatography you can use for the separation of inorganic components as well. Then it can be used for the purification of products of reaction in the laboratory as well as in the industry. So in a reaction, if you have different mixtures, different components in the reaction mixture, and if you want to separate that, you can use this technique. You can also use adsorption column chromatography. In our college, we use adsorption column chromatography to separate the reaction mixture. And then identification of organic and inorganic components of a mixture, you can use liquid partition column chromatography. So these are the applications of liquid partition column chromatography. So here what I have given here is the picture of high performance liquid chromatography, HPLC, that we will discuss in a later stage, not in this lecture, but this is an example for liquid partition column chromatography. So that is high performance liquid chromatography. So high performance or high pressure liquid chromatography, that means you are applying high pressure so that the separation is very fast. So this is an diagrammatic representation of high performance liquid chromatography. So don't worry about all these steps. So just focus on this column. So excuse me. So you are injecting your sample here, somewhere here. Then this is the this is the mobile phase. You can see two bottles. So sometimes the mobile phase, I told you, is a mixture of two liquids, 2 or 3, up to your samples. So that depends on the components in your mixture. So the nature of your components, you have to decide what is better for us as mobile phase. So in this picture, for example, we have two liquids. So the system, and you can feed here what is the combination of these two liquids. So automatically the system will take accordingly. So that mobile phase will carry your sample through here, and this is your column. So this is the enlarged view of a column. So this is a steel column, you can see that, and inside the inner walls of this column is either coated with your mobile phase liquid, or your mobile phase is supported on a solid which is coated on the inner walls of this column. And when your mixture is going through this column, that will get separated based on the difference in partition coefficient, and at the end you will use some detector to detect the signal, or detect which compound comes first, kind of stuff, that you will get the data from here in the computer. So this high performance liquid chromatography is an example for liquid partition column chromatography. So these are the two chromatography techniques which we discuss in this lecture. So thanks for listening to the lecture.